Scientists already have the DNA. They already have the technology. The question isn't if anymore. It's not if one of these animals comes back. It's which one comes back first. And whether we'll actually be able to control what we've built once it's standing in front of us. Right now, in labs from Dallas to Melbourne, teams are splicing ancient genomes into living cells, coaxing extinct traits back into modern bodies, and racing toward birth dates they've already put on the calendar. We've spent months digging through the peer-reviewed papers, the funding rounds, the leaked timelines, and the public statements from the scientists actually doing this work. What we found surprised us. Some of these projects are decades from anything real, held together by little more than a genome sequence and a hopeful press release. One of them has a due date circled in ink. And by the end of this video, we'll show you the most advanced and most dangerous attempt yet. A project that doesn't just want to resurrect the past. It wants to build something that never existed at all, and call it de-extinction anyway. So here's how we ranked them. 8 extinct species, counted down from the most speculative to the most advanced. From theory to funded research, to an animal that already took its first breath. Number 8 starts with a stripe pattern that just stops. Picture a horse-striped animal grazing the plains outside Cape Town in the 1870s, except only the front half of its body is striped. From the shoulders back, the stripes fade into a smooth chocolate-brown coat, like the paint bucket ran dry halfway through the job. That's a quagga. Not a separate species, a subspecies of the plains zebra, hunted by Dutch colonial farmers for meat, hide, and to protect grazing land for livestock. The last wild quagga was shot in the 1870s. The very last one on Earth, a mare, died alone in an Amsterdam zoo enclosure on August 12, 1883. Nobody in the zoo even realized she was the last of her kind. Here's the strange part. The quagga never really left the gene pool. It was a zebra with a mutation, not a different animal entirely. So in 1987, South African taxidermist Reinhold Rau launched the Quagga Project, selectively breeding plains zebras that already carried reduced striping genes, generation after generation, chasing the coat pattern back into existence. Therefore, this isn't cloning, it isn't CRISPR. It's old-fashioned selective breeding, done with a single goal: recreate the look, and maybe the genetics, of an animal that vanished from the record books. Here's where it gets strange. By the 4th generation, some of the Rau quaggas grazing in reserves like Elandsburg and Vrolijkheid in South Africa look almost indistinguishable from 19th-century quagga specimens sitting in museum drawers a continent away. And there's a fitting bit of history buried in this story. The quagga was actually the first extinct animal ever to have its DNA sequenced at all, back in 1984, from a 140-year-old museum skin. That single study effectively launched the entire field of ancient DNA research. Everything on the rest of this list, in a way, traces back to a quagga in a jar. Skeptics push back hard, though. Genetically, these are still plain zebras with a cosmetic trait dialed up. They carry none of the quagga's unique extinct lineage DNA, because that DNA, strictly speaking, never disappeared. It was hiding in plain sight the whole time, buried in the recessive genes of an animal that was never endangered in the first place. So is this de-extinction, or just very patient, very selective zebra breeding? Even the scientists involved don't fully agree. We've started this list with an animal that maybe never really left. What comes next is an animal that came back, and then left again, in under 10 minutes. January 6, 2000. Ordesa National Park, in the Spanish Pyrenees. Rangers find the body of a wild goat named Celia, crushed under a fallen tree. She isn't just any goat. She is the last bucardo, the Pyrenean ibex, on the planet. A subspecies of wild goat that once ranged across the mountains between Spain and France, driven to the edge by centuries of trophy hunting, poaching, and competition with domestic livestock for the same steep, grassy terrain. Therefore, when Celia died, the bucardo, as a living population, died with her. But scientists had already taken a small skin sample from her ear years earlier and frozen it in liquid nitrogen, a biological insurance policy nobody expected to need so soon. In 2003, a Spanish-French research team used that frozen tissue in a cloning attempt, transferring bucardo DNA into hollowed-out domestic goat egg cells, implanting the resulting embryos into surrogate goats. Out of 208 attempts, One single kid was born alive on July 30th, 2003. For a few minutes, the bucardo existed again on Earth. Then it died, a fatal lung defect, an extra lobe that made breathing impossible. It became, officially, the first species in history to go extinct twice. Here's the twist. For the scientific community, those 10 minutes were proof of concept. It showed that tissue frozen from an animal already gone could, in principle, be turned back into a living, breathing body, using nothing more exotic than a nucleus, a hollowed-out egg cell, and a surrogate species willing to carry it. That single kid became the blueprint every de-extinction project since has quietly built on, from the passenger pigeon labs to the mammoth labs in Dallas. Still, the failure rate tells its own story. Out of 208 attempts, only 7 embryos developed far enough to be implanted. Only 1 survived to birth. And that one died within minutes, its lungs choked by a defect scientists still don't fully understand. If this is the technology holding the future of extinct species in its hands are still shaking, 20 years later. The bucardo proved resurrection was technically possible. The next case on our list proves that having the DNA is only step 1. You still need somewhere for billions of missing bodies to come from. Imagine a flock so massive it blots out the sun for 3 straight days. Eyewitnesses in the 1800s described exactly that. Passenger pigeons migrating in numbers estimated in the billions, the single most abundant bird in North America, maybe on the planet. Then in barely 50 years, industrial-scale hunting and mass deforestation wiped them out entirely. The very last passenger pigeon, a female named Martha, died in her cage at the Cincinnati Zoo on September 1, 1914. From billions to 0, inside one human lifetime. The nonprofit Revive and Restore, co-founded by Stuart Brand, launched the Great Passenger Pigeon Comeback, led by biologist Ben Novak, sequencing the species' genome from preserved museum specimens and comparing it against its closest living relative, the band-tailed pigeon. So the plan isn't cloning. There's no living cell to clone. It's precision gene editing, rewriting band-tailed pigeon DNA piece by piece to carry passenger pigeon traits. Here's the catch. Researchers have built a strikingly complete genetic roadmap, arguably one of the most detailed extinct species genomes ever assembled, right down to the specific mutations that made the bird migrate, flock, and nest the way it did. But turning a map into a living chick is a different problem entirely. Bird reproduction doesn't work like mammal reproduction. There's no simple embryo transfer, no surrogate womb waiting to carry the pregnancy. Scientists need to edit primordial germ cells, the precursor cells that will eventually become sperm and eggs, and reintroduce them into a host embryo, a technique still being refined for this exact species combination. In birds, this specific. Confirmed genome, yes. Confirmed hatchling, still nowhere in sight, and no public date attached to one. So we've gone from billions of birds blotting out the sun to a single, painstaking cellular procedure that hasn't been proven to work yet. We've gone from an animal that essentially still exists, to one that vanished in minutes, to one that vanished by the billions with no living relative close enough to clone. Next, the gap between extinct and recoverable gets even stranger, because this next animal's closest living cousin can fit in the palm of your hand. Hobart Zoo, Tasmania, 1936. A striped dog-like marsupial paces its enclosure alone. Locals called it the Tasmanian tiger. Zookeepers called it Benjamin. Nobody at the time realized he was the last of his kind. Thylacines had already been hunted to collapse under a government bounty program that paid hunters for every carcass, blamed, mostly unfairly, for livestock kills that were probably wild dogs. Benjamin died in September 1936, and with him, an apex predator that had roamed Australia and Tasmania for millions of years, disappeared for good. Here's the scale problem. The thylacine stretched over a meter long, built like a cross between a wolf and a tiger. Its closest living genetic relative is the fat-tailed dunnart, a marsupial roughly the size of a mouse. Using it to rebuild a thylacine is a little like trying to build a canoe using a matchbox as your blueprint for scale. Still, in partnership with the University of Melbourne, Colossal Biosciences reconstructed close to 99.9% of the thylacine's genome from an ethanol-preserved pouch joey specimen, one of the most complete ancient marsupial genomes ever assembled. Therefore, the genetic blueprint exists in extraordinary detail. Here's where it gets strange. Colossal has publicly claimed real progress editing Dunnart stem cells toward thylacine traits, treating this as one of their flagship projects alongside the mammoth and the dodo. And the company has even formed a dedicated advisory committee to plan the animal's eventual return to Tasmania. But independent marsupial biologists point out something the press releases tend to gloss over: marsupial reproduction, especially the pouch-rearing stage where a joey the size of a jelly bean has to crawl, unaided, into its mother's pouch and nurse for months, is barely understood in a laboratory setting. No one has ever grown an artificial marsupial womb capable of carrying an animal this size, and no one has ever scaled a Dunnart-sized embryo up into anything resembling a meter-long predator. A stunning genome, a living animal that may still be, at best, a distant hope. The thylacine shows us a genome without a body. Our next case flips that entirely, a body preserved so well in ice, scientists can practically examine its last meal. Yakutia, Siberia, 2014. Local reindeer herders stumble across something extraordinary melting out of the permafrost. The frozen carcass of a baby woolly rhinoceros, later nicknamed Sasha, roughly 10,000 years old, with skin, fur, and internal organs still largely intact. Picture a rhino built like a linebacker in a full-body coat of shag carpet, with a distinctive fatty hump over its shoulders, bigger than a modern rhino, built to survive brutal Ice Age winters across Siberia and northern Europe. Climate change and human hunting pressure closed in on the species roughly 14,000 years ago, and therefore, unlike most Ice Age giants, the woolly rhino left behind bodies cold enough and complete enough to still contain usable soft tissue. That's the twist here. While most extinct species leave scientists working from fragments and guesswork, permafrost specimens like Sasha hand researchers unusually well-preserved DNA, arguably better preserved than the woolly mammoths. Colossal Biosciences has folded the woolly rhino into its research pipeline, using the critically endangered Sumatran rhino, genetically the closest living relative, as the template for comparison and, eventually, as a potential genetic base. But here's the catch nobody likes to say out loud. There are fewer than 50 Sumatran rhinos left alive on Earth. So the living relative this entire project depends on is itself teetering on the edge of extinction. Is it responsible science to lean on an endangered species biology to chase a woolly rhino down a research rabbit hole? Or should every dollar and every embryo be spent protecting the 50 that are still breathing right now? Even inside the conservation community, that's not a settled question. We've now seen ancient tissue, reconstructed genomes, and a genetic donor species that's endangered itself. Our next case takes all of that and adds a company with $10 billion in funding and an actual due date. Picture something the size of a small house, draped head to toe in shaggy brown hair, tusks curving out longer than a compact car is wide. That's the woolly mammoth, an animal that dominated the Ice Age tundra for hundreds of thousands of years before climate shifts and human hunting pressure squeezed the species down to a final holdout population on Russia's remote Wrangel Island, which finally blinked out roughly 4,000 years ago, around the time Egyptians were already building monuments that still stand today. Colossal Biosciences, founded in 2021 by Harvard geneticist George Church and entrepreneur Ben Lamb, has assembled over 50 mammoth genomes recovered from Arctic permafrost, spanning more than 1 million years of evolutionary history. Their method: edit mammoth traits, cold-tolerant blood, small ears, thick subcutaneous fat, shaggy hair, directly into the genome of the mammoth's closest living relative, the Asian elephant, which shares roughly 99.6% of its DNA. So this isn't cloning a mammoth from scratch. It's engineering what scientists sometimes call a mammoth proxy, an elephant carrying enough edited mammoth genes to survive, look, and behave like one. Here's the twist that separates this project from everything else on our list. In 2025, Colossal actually showed the pipeline working, not in an elephant yet, but in mice. Genetically engineered woolly mice with cold-tolerant traits, golden brown coats and curly whiskers, built as a small, fast proof of concept before attempting the real thing in a 2-year elephant pregnancy. CEO Ben Lamb has stated the company remains on track for its first mammoth calf by the end of 2028. But still, no elephant has ever been successfully cloned by anyone, ever. Editing thousands of mammoth-specific genes into a living, viable elephant embryo, then carrying it through a 22-month gestation inside a surrogate, is a feat nobody on Earth has pulled off. And even Colossal's own Chief Science Officer Beth Shapiro has been careful to say the result won't be genetically identical to a real mammoth, just functionally, ecologically similar. A near mammoth, close enough to fool the eye maybe. Close enough to fool the tundra? Nobody actually knows. The mammoth has a due date. Our next case has something even rarer. A nearly complete genetic blueprint, reconstructed from a single one-of-a-kind museum specimen. Mauritius, late 1600s. Dutch sailors describe a flightless turkey-sized bird waddling fearlessly toward them, an animal that evolved on an island with no natural predators for millions of years, and therefore never developed the instinct, or the wings, to run. By 1681, it was gone. Not just hunted by sailors, but wiped out by the rats, pigs, and monkeys those same ships accidentally introduced, which raided dodo nests faster than the birds could rebuild their population. For centuries, almost nothing physical of the dodo survived, until researchers turned to the Oxford dodo, a specimen at Oxford University's Natural History Museum that still contains preserved soft tissue, one of the only such remains on Earth. In 2022, a team reconstructed a near-complete dodo genome from it, comparing it against the bird's closest living relative, the Nicobar pigeon, a striking, iridescent bird found across parts of Southeast Asia. Colossal Biosciences, with Dr. Beth Shapiro's team involved, has taken on the project, aiming to edit Nicobar pigeon DNA toward dodo traits. Here's where it gets strange. This genome is unusually complete, arguably one of the clearest genetic pictures of any extinct bird on record. Therefore, on paper, the dodo looks like one of the more achievable projects on this entire list. But the counterpoint lands hard, just like it did with the passenger pigeon. Bird cloning has never been done successfully, anywhere, on any species, and the primordial germ cell technique this project depends on is still unproven at this scale. A gorgeous blueprint. No confirmed timeline for a hatchling. No promise that the process even works the way researchers hope it will. We've climbed from a striped zebra to a genome pulled off a museum shelf. Every project so far has chased something that used to exist. What's coming at number 1 doesn't just chase the past. It asks how far scientists are willing to go once close enough becomes good enough to call something back from the dead. October 1st, 2024. Deep inside a secret, 2,000-acre preserve somewhere in the United States, 2 wolf pups are born by planned cesarean section. Snow-white fur, oversized shoulders and jaws. A howl their handlers say sounds different from any living wolf. Colossal Biosciences names them Romulus and Remus. A third pup, Khaleesi, follows in January 2025. In April 2025, the company announces to the world that it has done something no one in human history has ever done, brought an extinct species back to life. The dire wolf, Ice Age apex predator, extinct roughly 10,000 years ago, made famous decades later by a hit fantasy TV show, is declared restored. Here's the catch, and it's a big one. To create these pups, Colossal didn't reconstruct a dire wolf genome from scratch. They took a modern gray wolf and made just 20 precise edits across 14 genes, out of roughly 19,000 genes total in the wolf genome. That's a fraction so small that geneticist Pontus Skoglund of the Francis Crick Institute joked the pups were, optimistically, 100,000th dire wolf. Even Colossal's own chief scientist, Beth Shapiro, eventually clarified the truth in plainer terms. These are gray wolves, with about 20 edits, cloned. Paleogeneticists like Nick Rollins at the University of Otago point out that dire wolves and gray wolves actually split apart millions of years ago, genetically closer to jackals than to the wolves roaming forests today. So the animals standing in that preserve aren't dire wolves reborn. They're something new, wearing a costume built from 20 genetic edits and a marketing campaign. And that, here at number 1, is the real twist of this entire video. Every project we've covered so far has been chasing a return, trying to rebuild something the world already lost. But the dire wolf shows the industry's actual capability, right now today. Not perfect resurrection, but the ability to engineer any trait into any living animal, and call the result whatever you want. If 20 edits are enough to convince the public, and half the scientific press, that an extinct predator walks the earth again, what happens when a company decides to stop imitating the past and starts designing something that never existed at all? A predator built from scratch. A de-extinct species that's really just a new one, invented, patented, and released. Some scientists still defend the framework. Shapiro has argued that if an animal looks, behaves, and fills the ecological role of the original species closely enough, calling it a functional de-extinction is scientifically defensible. A practical definition for an impossible task. Critics call that argument exactly backwards. A justification built after the headlines, not before them. One that quietly erases the line between bringing something back and simply making something up. 8 animals, 8 timelines. From a zebra that never really vanished to a wolf that never really returned. Scientists already have the DNA. They already have the tools to cut, splice, and rewrite it. What they don't have, what none of us have, is a clear answer for what happens after the cage door opens on something new. So maybe the real question was never whether we could bring these animals back. Maybe it's whether the thing that walks out of that lab still deserves the name we gave it in the first place. If you want to know which extinct species almost made this list, and the one de-extinction project that already went catastrophically wrong, that's next. Subscribe and we'll see you there. See you.